we're going to talk about the microcytic anemias. First is the iron deficiency anemia. So in iron deficiency anemia, we have decreased iron that is going to cause problems, or anemia actually. Now in iron metabolism, so first in iron deficiency anemia, we're going to do iron metabolism. That is what is important. So I'll give you one overview of what it looks like that we have to cover. Looks intimidating, but it's actually pretty easy. I'm gonna break it down. So what we're gonna do right here, I'm gonna draw a small scheme right here. I'm gonna start from, the environment. Okay, we take iron from the environment and then we eat it, right? And then we eat it and it goes to our stomach. Then from our stomach it is gonna go to our duodenum, and from the duodenum it is going to get absorbed into the blood, and from the blood it is going to be transported to different location, distributed. It could distribute into liver, to erythropoietic tissues where you make blood cells, erythropoietic tissues, and sometimes in muscles and many other areas for utilization wherever it is needed. And so that is, the whole thing. But of course, there are details. And because iron is, only, kind of like defecated, or you could just, released either by menstruation, one, or the other is epithelial shedding. There is no other way to get rid of iron. So that is why iron is tightly regulated, and liver produces a protein that I'm going to tell you in the future in this video, is that regulates this whole iron metabolism. So the food source. So now we start with the environment, the food sources. So the first food source is the animal source. That is heme iron. We call it the heme iron. And heme iron is Fe+2. It is called as the ferrous form. Okay, ferrous form. So I would like to remember it like the way that, because it's 2, so 2 has O in it, and ferrous has O in it. So that's how I remember the Fe+2 is ferrous. And Fe+3 is ferric. And that is from plant sources or non-heme iron. We call it the non-heme iron. And other than the food sources, we can also have iron from daily RBC turnover that are senescent RBCs. So it will go into the stomach. So in stomach, we have hydrochloric acid from the gastric juice. So what it does is that it converts all the Fe+3 into Fe+2, right? Now, why is that important? Because in duodenum we have enterocytes. In duodenum we have enterocytes. And enterocytes, in the enterocytes, Fe+3 cannot enter. So we need to convert all Fe+3 into Fe+2 in order for it to enter enterocytes. Now, half of it has already been converted by HCl, but still there might be some that might be left. So on the brush border of enterocytes, we have cytochrome B, or it can be ferroreductase or DCTYB. These are the enzymes that convert Fe+3 into Fe+2. After Fe+2 is generated, there are two transporters. One is the heme carrier protein, and the other is called as DMT1, divalent metal transporter 1. And these are doors in which the iron is going to leave the intestinal lumen and enter the enterocytes. And inside the enterocytes, now it can be stored. It can either be stored in the form of mucosal ferritin or hemosiderin. Ferritin is a short-term storage of iron, while hemosiderin is a long-term storage of the iron. Now, because the iron is stored in these enterocytes, and intestinal epithelium is generating almost daily, and that is how we can shed this iron, and that's how we can get rid of iron from the body through feces, because of the intestinal shedding. All right. Now, if our blood levels of iron is, decreased, what can happen is that mucosal ferritin, it can, ferritin can leave iron, and iron can leave via the ferroportin 1. Here, it's ferroportin 1 is basically doors. So these are the doors from which iron can leave the enterocytes and now enter blood. So again, I'll write it in simple: iron in lumen. Okay, I'll just add iron plus 2. Okay, it comes in to the enterocyte via DMT1. That's more important to remember, DMT1. And in the enterocyte, it is in form of ferritin, and then it will go to blood, Fe plus 2, and it is going to go via ferroportin 1. So this is important. This is the crux that you need to remember. But in blood, we cannot leave iron alone. So iron cannot be left alone because of Fenton reaction. So what is a Fenton reaction? Fenton reaction is that the free iron forms free radicals, and that damages tissue. So that's actually Fenton reaction. So what we do is that there is another protein called as the transferrin. But the problem with transferrin is it cannot take Fe plus 2. So we have two more proteins or enzymes. One is hapestin, and the other is ceruloplasmin, that converts Fe plus 2 into Fe plus 3. And that is then carried by transferrin right here. And now our absorption is completed. This is iron absorption. Now we do iron transport. Iron is transported via transferrin, and then it is distributed to erythropoiesis, muscles, and liver. Now we talk about the regulation of iron. That is, liver produces hepcidin. Now, if there are more, the iron is increased, or the iron stores in liver are increased, then what is going to happen is that hepcidin will be produced. And what hepcidin does is that it gives sequesters ferroportin, so it closes this door, and therefore iron cannot enter blood, and it stays in its mucosal ferritin form, right? And another thing it does is that it goes to macrophages and inhibits macrophages from producing iron. That is how it decreases iron in the blood. And of course, metryptase, another protein, it decreases hepcidin. So that is going to increase iron in the blood. Now, of course, there is more to different regulatory factors that can affect iron, but that is a discussion for another day. We're going to do iron deficiency now. So for iron studies, we have certain labs. We do serum iron, that tells us how much iron is in the blood. And then we do, the next is total iron binding capacity, or TIBC, that tells us number of transferrin. It only tells us number of transferrin. Then we have percent saturation, that tells us how much transferrin is bound with iron. So it's important to differentiate between TIBC and percent sat. Then we have serum ferritin. It tells us the iron stores in liver and macrophages, not the intestines. Now, in iron deficiency anemia, what are you going to find? You're going to find microcytic and hypochromic RBCs. Now, I already told you how they are microcytic because they have less mean corpuscular volume, because we have less iron, and less iron means less heme, and less heme means less HB, and because there's less HB altogether, that is going to, decrease the cell size, because we know that during erythropoiesis the cell size increases because of accumulation of hemoglobin. And why it is hypochromic? We know that the color of RBC is imparted by iron in hemoglobin. So when there is no iron, of course there is no color, so hypochromic. And then there is going to be decreased ferritin and increased TIBC. Now there's going to be decreased ferritin because the iron storage pools are decreasing. I'll tell you further in the stages of iron deficiency, and I'm going to detail the other labs there. So first is the initial state, then we have the second, third, and fourth stage. So in initial stage, only the iron stores are going to get depleted. So you see right here, ferritin decreases, but the TIBC increases, or it remains normal, you could say, in blood, because there is sufficient iron in the blood. That is why TIBC is normal. Only the storage pools are depleting. Then in the second stage, serum iron is now depleted. First the stores were finished, and now the serum iron is depleted. That is why the SATs also decrease. The percentage saturation of transferrin also decreases because there is no iron in the blood as well. Now in the third stage, there's going to be normocytic anemia, because bone marrow is going to form less RBCs, but they're going to be of normal size. Now, in the fourth stage, in the final stage, there is now going to be the microcytic anemia, because not only bone marrow makes less RBCs, but also makes small RBCs. That is how MCV is reduced, and hence microcytic anemia. Now, there are going to be certain causes of iron deficiency anemia, and it can be at any age. So in infants, it could be due to breast milk. And we know that we start weaning at 6 months. So if the weaning is not started at the proper time at 6 months, and only breast milk is fed to infants, then they're going to have iron deficiency anemia. And in child, of course, it's going to be poor diet. In adults, in females, either it's going to be menorrhagia or in pregnancy because of a lot of blood loss, and in pregnancy, of course, the demand is increased. And in the males, it's going to be peptic ulcer disease, and in peptic ulcer disease, we know that there is achlorhydria, and in achlorhydria, we have decreased HCl. And of course, we need HCl to convert Fe+3 into Fe+2. And then we have elderlies. We can have polyps or carcinoma, and of course, hookworms like Nectaris americanus, or the other hookworms. There could be malnutrition, there could be malabsorption like celiac disease or sprue, and gastrectomy, as I already told about, because of HCl. And then finally, we have Plummer-Vinson syndrome, which is basically a syndrome which has a triad of esophageal webs, iron deficiency anemia, and glossitis. And that is it for iron deficiency anemia.